because I I decided that this time you will be tired enough to hear a story instead of some very complex issues. It was so inspiring talks that what I wanted to tell you today is kind of history. It's pity that I cannot just show you the abstract of my presentation. You can find it. What I wanted to tell actually that a story about the paper DNA multiple codes that my close friend Philip Bucher, supervisor Edward Trifonov, published about 35 years ago, and after that it was other publications from him about DNA codes. And so I wanted to tell you the story about this publication and my impressions at that time, today, what we can learn from this, and so on. So it's kind of history. At that time, the publication was in '89. I was his PhD student, and my background is from computer science. So I came to computational biology from computer science, and Edward Trifonov, in his time, he came to molecular biology from biophysics. So we have a little bit different backgrounds, and when he was a physicist, he was involved in kind of coding theory. So they're absolutely different codes, of course. And so for both of us, the word code had very different meanings. But I would say that if I had internet at that time, and in '89, of course, we didn't have internet, I could do it what I did much later. Just I wrote in Google code definition, and I found many different definitions of this word. So this, when you use the word code, you should understand that it's for scientists from different areas, it will have absolutely different meaning. But for me, it was interesting to understand what Ed Trifonov means when he uses the word code. And for him, it was the first reason to use this word was that it was genetic code, and he wanted to show that there are different genetic codes in genetic sequences. Not only that what we call genetic code, so we better call it triplet code, that it's coding of protein that are different genetic codes. He meant it that people that don't see what is the meaning of non-coding sequence. Coding sequence, it's coding for protein, and non-coding sequence is something that at that time many people called junk DNA. So it was very important to show that there are different things in the sequence. And so this was the first motif, and he introduced his definition of code, of sequence code. And it was something like any pattern, as any sequence pattern, or even bias in sequence with function. Of course, and the word function here it's very close to the word meaning that we use today, especially Marcello when he discusses the codes. So it was a pattern with function, with meaning. And pattern it's very basic term that it's everything that you can somehow describe. Okay, so this is for me it was something that it's really I didn't like. So another explanation, what does it mean? It was that that in DNA text you can find many different messages. And these messages are instructions. So the code in this when we say pattern with meaning, it's a necessary instruction for something. So instruction, it's of course, it's much closer to understanding. For me, it was at that period, but still, just to have, for me or for other readers to understand what it's talking about. So Edward Trifonov in his paper brought a few examples of codes, and his involvement is this sequence analysis started when he looked for certain periodicities in DNA sequences, and for him it was to finding this kind of periodicity. It was related to nucleosome positioning code. Maybe he didn't call it at that period code, but it was nucleosome positioning. So sometimes this code, people call it chromatin code. Sometimes chromatin code, they mean different, but anyway, if you try today to look in the literature chromatin code or nucleosome positioning code, for my great surprising, you'll find very, very recent publications as well. Even that many people, including Edward Trifonov, several times published the papers that they called, as it was the name of one Ed Trifonov's paper, the final cracking of nucleosome positioning code. So it means people published many times that they finally solved the problem how the certain pattern in sequence rules where nucleosomes should be positioned. In spite of this, people continue to publish papers, and from this point of view, this is still a very open question. So here, when we go back to the paper, so we find this example of nucleosome positioning code, and we ask the question today, what we can say about this part of the paper. So we see that actually the question whether such a code exists from the point of view of Trifonov code by the name that it's certain pattern, we cannot be sure. Moreover, we have a lot of evidence that if there is such a pattern, it's not one pattern, it's quite a few rather different patterns. So we should say set of patterns without real understanding today why one pattern will be preferable for among other patterns. So it means that this problem is still very open. But the second problem with this code is a problem that was mentioned by Marcello many years ago, that from his point of view, this cannot be called code, nucleosome positioning code, because it's a clear physical appearance of dinucleotides. It's not just this kind of selection of dinucleotides that may be arbitrary. It cannot be arbitrary because they have a very strong physical characteristics. And indeed, if we'll look for recent publications, you continue to see many physicists that really don't know many about nucleosomes, but they continue to describe the physical characteristics of DNA that maybe will bring certain ruling of nucleosome positioning. So it was the first interesting for me about this nucleosome positioning code. But the other characteristic of this code is somehow related to the previous talk of Philip, because when I remember all this history of nucleosome positioning code, I remember the huge disappointment that we got about the universality of this nucleosome positioning code. According to all science, it should be some universal code because DNA is the same DNA everywhere, and nucleosome, it means octamers, histones all together, they are highly conserved. It's among the most conserved proteins in evolution. And still, so we could expect that we can find the same. Oh, I think that it's the second abstract. Mm-hmm. Next. Mm-hmm. Yeah. Okay, so thank you. I think that I will be able. Okay. Okay, great. Here I discovered for myself that it will be rather good reason to write a paper that still I continue to think that maybe it's worthwhile to write about universality of codes. When Ed Trifonov started to his studies of different codes, he was sure that he will find more or less universal codes. And when we look back today on all these discovered codes, we see that practically nothing is universal except of the so-called genetic code, this triplet code, that it's really universal. But all other codes that we see that are very various. It means that, first of all, practically impossible to find something that's in common between prokaryotes and eukaryotes. But in eukaryotes, we can see that it's so huge variety of different patterns that we see for transcription or even for translation when we try to describe it in the language of codes, that we must be disappointed with this approach of universality. It means that what Philip told us, it's a great surprise, because we don't know what kind of code is in these ultra-conserved elements, but if is this code that it's conserved among different organisms, so different species, it should be very interesting exclusion in the rules that we have everything is not universal. Suddenly we find something universal. So it's awesome. So it was about this is called chromatin code. The story with translation framing code, it's very interesting. The idea of this came that Edward found that there is so-called 3BP periodicity in coding sequences. And as a person that came from coding theory to molecular biology, his first idea was that it's kind of error-correcting code, that because ribosome, when it makes translation, it should keep the right frame to do translation. And this periodicity helps to do it, and he even in very interesting details describes how it could be. So it was the translation framing code, that it was a hypothesis that I like very much, because it's very beautiful hypothesis from many points of view. But unfortunately, even may I get some water? It's difficult. Even 35 years after that, we still don't have real experimental justification of this hypothesis. Even that it's very reasonable what it was in this framing code. still we don't have a real experimental justification. Okay. After that, we have so-called shape code. Shape code, it's related to curved DNA. So if you Google in today it's curved DNA, so you'll find many papers. And rarely the shape code it's mentioned there, but it's again, if we said that nucleosome positioning code, it's not a code from the point of view of code biology. So shape code, it's nothing to do like this, because we have a certain physical characteristic of DNA that brings it that without any forces, this DNA will have not a linear axis, but it's curved axis. So it's very interesting. But it's not a code from our point of view, and whether it may be considered like a code from Trifon's point of view, it's also very questionable, even less. But from another point of view, if we will consider that there are some proteins that make so-called indirect binding to DNA. It means that instead of kind of reading the sequence to find the site to bind to DNA, they somehow recognize the shape of DNA to bind to certain shape. There's some of transcription factors, for instance, they probably have this property. At least there are many publications on this. From this point of view, it's already certainly kind of code, because we have some instruction to protein to bind to this kind of structure, and this structure is dependent on sequence. So maybe shape code from this point of view, it could be a good candidate to be a real code. Loop code, this even I don't want to discuss it now, because even about the other stories, I said that it's have no real justification, experimental verification, and so on. So this loop code is even less than this. But for me, it's very interesting that I'm the student and the admirer of Trifonov, and I is a kind of critic here. But for me, it was very interesting to understand this paper that is practically all these examples are not good examples of code. This paper has many citations that many people refer to this paper, and I'm ready to claim that it is really very important and very inspiring paper. So it means that for me it's interesting problem how you can write a paper that it is pure biological point of view. All the discoveries are, let's say, questionable, but it's a paper that has a big impact on development of a certain direction in science and so on. So this is another very interesting aspect. I said already there are two. The one, it's universality of codes, that we can see that it's appeared not so universal. And the second, it's about what makes a paper so inspiring and important. Okay, so what else was in this paper? It was not really about codes, it was about what we call today, or in that way, it's Trifonov introduced the term DNA linguistics. So when we try to study about sequences from this linguistic point of view. So I expected that maybe it will be some connection to the biolinguistics, but not. I see that it's not the same. And this is possible to say branch of code biology, sequence biology, it probably will not contribute something to answer to your very interesting questions. Okay. So this is what I wanted to tell you as a story, and I decided not to make the pictures to show you what every code, how it's presented, and so on. I just wanted to say that among the papers of Edward Trifonov, on the first place, the papers that are related to curved DNA. And the second place, maybe this is very special paper that he wrote together with his PhD student Philip Bucher about the promoters. But it was kind of a very special publication, mainly because of personality of Philip, because it was not typical for Edward Trifonov. The third kind of, it was in nucleosome positioning, a lot of papers. But after that, as a most cited paper, it was on the bunch of papers about multiple DNA codes. So for me, it's absolutely no doubt that it was important and inspiring paper, and this is the reason that I wanted to remind you about this paper. Maybe you will have a look at it as well. So, thank you.
Thank you, Professor Alexander. So we have time for a few questions, please.
So it's it's obviously important that this paper was one of the first factors for Marcello. I mean, he discovered this paper rather early and referred to it early. It has been a very important paper. So I have a question about the shape code. So again, the problem is that what we have here is a structural feature, a three-dimensional structural feature that is recognized by something else. Mm-hmm. But there's no adapter in between. So it doesn't really fit this definition of code, that it has to be a translation via an adapter. So it's more a direct recognition code, if it's a code. I would hesitate to call it a code.
Yeah, but Trifonov defined the code.
But I know, but the point is, so Professor BOLSHOY is saying that this could be a code in our terms, but it's not a code in our terms. Yeah. Okay, so just a comment, but...
Yeah. Okay.
Maybe I can refer directly to this. It may be part of the code. Whatever binds to this structural DNA element may bind or mediate to something third, and then we have a code.
Yes.
Yeah.
But then you need to show that thing that binds is the adapter that has an organic meanings that it translates into. That would be very interesting.
Sure.
Yeah.
Well, I just wanted to add the following. I think the question here is whether DNA structure is the only responsible biophysical basis of the nucleosome code, or whether the octamer is a potential adapter, which, has specific preferences, slightly different at different places. Now, in the latter case, it's simply a universal code because the octamers are always the same. But if one would be able to engineer an alternative octamer, which prefers somewhat different nucleosomes, then I would accept that it's a code in the sense that it has an adapter to it. So,
yeah, I wanted to say here that it's, thank you that you remind me. I wanted to put some attention of additional problem. The question is, if we want to answer the question whether nucleosome positioning code is a code from Trifonov's point of view or from Barbieri's point of view, we have to answer the question for us, what kind of tools we can use. Actually, I was a person that proposed at some point to use a multiple alignment as a tool to discover this pattern in DNA, and after that to use it. Many years after that, I came to the conclusion that my advice was wrong. That if we want to find this kind of pattern of code, multiple alignment is not good enough. So this is a very interesting question, and maybe I was right that time, maybe I'm right today, but anyway, this is an interesting question for code biology. What kind of tools we can use to find an answer whether the code exists here, and what shapes this code.
I've been working with this Trifonov concept of codes by several years, and we begin working on this free alignment problem. That means that we want to split and chop all the sequence in different lengths and try to see if we can find a pattern that helps you to difference different taxonomic groups. Mm-hmm. In this case, we found that we indeed can chop to a fixed length the sequence, and we can correlate to some phenotypic function and morphology traits. But in this case, the missing part is still the adapter, because we know that we have a structure or we have some syntax or of the sequence and some organic meaning, but we're still missing this part when we can validate that we connect both words. So I think there is a way to use Trifonov concept to turn it around in Barbieri concept of code, but we need to start finding the adapter. But certainly there is some
you want to say that 50 years of research is not enough. Yeah, so this in spite of many publications that we solved this problem completely, it's still unsolved problem. Yeah, I agree with.
Sorry, it is hard to put it in adapter terms, but I saw once a wonderful work of a scientist, actually a Portuguese scientist, that they will freeze the nucleus of the cell and slice up in 3 dimensions and look up which parts of the genome were close together, and this could help you find some sort of proximity and help to introduce this shape code, and if there is any relevance. But as Robert said previously, it could be part of the Eastern code.
No, but probably it's not a shape code, what you say, because if it's eukaryote, it's more chromatin code, because everything is in chromatin. It's a very complicated issue, because there are so many factors that make different parts of genome in close proximity. It's very interesting, very important problem, but it's not only a so-called chromatin code here, it's like this.
Well, it seems to me that you have to put this into an historical context. Mm-hmm. Trifonov mentioned about 7 or 8 codes, some of which have adapters and some of which do not have adapters. Remember that he started working in these papers in about 1987, 1989. He actually was one of the very first, was going to say, look, in the genome, in people, there are many codes, not just a few. This is his big contribution. The fact that then he defined a code as anything which is a function is another matter. We can discuss about that. But I would say the major contribution of Trifonov is that, remember, tell people, look, in the genome, there isn't just the genetic code, there are many codes overlapping that. So he had a great historical, that point, it has a great historical impact, historical value. The fact actually that he mentioned many codes, again, gives you an example that he mentioned he had an idea that there are many codes, many types of codes, to the point that he actually accepted a definition which could include most of them. So from this point of view, he was a pioneer. It was a real. The fact that now we distinguish different type of codes is a supplementary thing. I think his contribution should be recognized as one of the founding fathers of the multiplicity of codes in life. And about the definition, we can go on and restrict our meaning. But the point is that some of these codes have adapters, some have not. So we have to look at this from this more general point of view. Okay. So it was a great story.
Okay, so you're invited to write a paper. Why this paper was inspiring? What should be in the paper to become inspiring for decades? This is it's not biological, but it's philosophically or psychologically. It's a very interesting question.
Well, remember that he was working in a time at a time when even the genetic code was not considered a code. Mm-hmm. There was this hostility of the biologists to accept something which is arbitrary means. Mm-hmm. So at that time, in that context, to say there is not just one, not only the genetic code is a real one because you can prove it, but the fact that there are many codes and many different functions and so forth. That was, in that time, it was a great historical contribution. And then we can go down into details if you want to distinguish but yes, I have objection about giving the name code to some of them, but some others are certainly codes in the sense that we have. Anyway, to me, the great contribution of Trifonov was that this concept that there is a plurality of codes in the genome and so forth, and that doesn't, it doesn't diminish the fact that we can recognize different types and so forth.
I will pass your opinion to him.
Okay, yeah.
Thank you, Professor Alexander. Thank you very much.